Good morning, students. So, let's start with our revision part. We are revising now our proteins, right? We finished with the enzymes, amino acids, and now we will move on to proteins. As we know that proteins, they are made up of our amino acids. Proteins, they are made up of amino acids. And the word protein, who first time gave the word protein, that was Bergelius. Bergelius in 1838 he talked about the word protein, but this word was actually used by another scientist which is known as Mulder, G.J. Mulder in 1840. When we say structure of proteins, there are four major structures: primary, secondary, tertiary, and quaternary, right? So whenever either in the subjective question or in the objective questions, if any question is asked from this topic, that is structure of proteins, then you should go through primary, secondary, tertiary, and quaternary proteins with their functions. So we know that our proteins, they are made up of our micromolecules, which are amino acids. So due to different rearrangement, I will say, of amino acids, proteins, they have different types of structure. Like primary structure is a very common, where not very common, I will say, it is a very simple structure of a protein, in which linear sequence of amino acids is arranged. Basically, amino acids, they are arranged in a linear sequence, means amino acid 1, then it is joined with amino acid 2, then it is joined with third amino acid. In this way, there is a long linear chain is appeared in primary structure. When we say long linear chain, it means amino acids are joined from one end to the another end. So first end, or you can say left end of the protein, is known as N terminal, and the last end of the protein is known as C terminal. So primary structure of the protein, in which sequence of amino acid or amino acids are arranged in a linear sequence. Left end is known as N terminal. It is because it is represented by your first amino acid. Suppose your first amino acid is serine, then its N terminal will be our first left end, and last amino acid is glutamic acid. Its C terminal will represent last end. So first terminal in the primary structure is known as N terminal, and last end is known as C terminal. Now, so this is, suppose we have taken, like I have taken 4 amino acids: serine, cysteine, tyrosine, glutamic acid. So serine joined with the cysteine, then it is joined with the tyrosine, and it is joined with the glutamic acid with the help of peptide, bond. That is the major thing which you need to understand, that amino acid, when they are forming a polypeptide chain or a protein, they are joined with the help of CONH bond. What is that bond? It is peptide bond. So covalent connections of a protein, it is a linear, what do you learn from a primary structure? That many amino acids, they are joined with the peptide bond, and the nature of the protein, as well as how long it is, it depend on the number and nature of amino acids. So insulin, myoglobin, lysozyme, these all are our primary proteins. Usually we say that primary proteins or proteins which are having structure, primary structure, they are non-functional. But we have one major exception here, insulin. Insulin is primary in the structure, but it is functional in the nature. So this exception you have to write down separately. Now move to our second structure of the protein, which is known as secondary structure. Till now your sequence was linear. Now we have started folding this structure, right? You have started folding this linear chain. So folding of a linear polypeptide chain into a coiled structure is known as secondary structure. Suppose like in your hair, keratin of your hair, that is your protein which is showing secondary structure. Nails have a keratin that is also shows secondary structure. Fur, claws, feathers, they all have proteins with the secondary structure. There are two secondary structures which are known to us, that is, one is like this sequence, that is alpha helix. Another is like the pleats, like जैसे गर्ल्स की स्कर्ट्स में जो प्लीट्स होती हैं, so that is called beta plated sheet. So here first let's understand this alpha helix. As you can see here, these are our amino acids and they are joined with peptide bond. So these amino acids, they are joined with peptide bond. Now when this polypeptide chain is curling up, it's folding up, then there will be the interaction between these two end. Suppose like this was my linear chain and now first fold occurred. So last amino acid will interact with first and first will interact with other. So in this way there is the interaction between the amino acids from last to first end, and that interaction is with the help of हाइड्रोजन बॉन्ड्स. देखो जो टू अमाइनो एसिड ज्वाइन कर रहे हैं एक दूसरे से, दैट इज विद द peptide bond. But when one amino acid, or you can say this amino acid, is interacting with the first amino acid, then the bond which is forming here is hydrogen bond. This is alpha helix structure. The another structure which is represented in the secondary structure, that is the beta pleated sheet. It is also same linear sequence में amino acids, they are interacting with the help of peptide bond. But now this is one complete polypeptide chain, this is second polypeptide chain. So these two interact with the help of attach with others, that is with the hydrogen bonds. Usually silk fibers, they have basically silk protein, which we say they are having this beta pleated sheath. And your keratin of hair, claws, feathers, the protein present in them, they are usually alpha helix. Tertiary structure, tertiary means more folding. Now folding of long protein chains. What should I call these long protein chains? Means polypeptide chains. Long protein chains means polypeptide chains. And suppose now when these one polypeptide chain, second polypeptide chain, third polypeptide chain, so when they appear like a ball, woolen ball, and they actually responsible for 3D structure of a protein. So who is responsible? Which structure is actually responsible for 3D view? That is our tertiary structure, and mostly globular proteins. I will tell you one mnemonics how to remember these globular proteins. These are our they show which kind of structure? Tertiary structure. So now here you can see that there I have made 3 polypeptide chain, blue color, green color, red color. This end is N terminal. Here second polypeptide chain, and here end is our OH group is there. C terminal basically. So now if suppose this is the active site of this protein, so when any substrate has to bind here, then who is going to bind, which can form a bond between with this OH and NH2. If any structure which cannot form bond with NH2 and OH, that will not bind here. So OH and NH2 is bonding. So bonding with OH and NH2 is very important for the substrate. So this place is known as active site, and only specific substrates can bind to these active sites. Same goes for these area. If any substrate has bonding for this place, only then can come here. So it means tertiary proteins, they are very specific for their active sites, and if they are very specific, it means they perform specific functions. So tertiary proteins are the one which are very specific in their functional roles. Bond which are going to play one major role, bond is our peptide bond. Another bond is hydrogen bond. But if suppose your amino acids, they are sulfur, then there will be formation of disulfide bonds also. And then because there is the hydrogen bond is also involved, and C terminal is there, so ionic bonding is also very familiar here. So peptide bond, hydrogen bond, disulfide bond, ionic bond, these are our specialty of tertiary and quaternary structure. Quaternary means 4 polypeptide chains, which is the special feature of our hemoglobin. Hemoglobin shows which structure? Quaternary structure with the 4 polypeptide chains, 2 alpha and 2 beta, right? So basically quaternary is also specifically functional. Protein part, globin part, I have written here globin part. It means protein part of hemoglobin is having 4 polypeptide chains, 2 alpha, 2 beta, and it shows which structure? Quaternary structure. Bond is involved are the same: peptide bond, van der Waals forces, hydrogen bond, ionic bond, disulfide bonds. These bonds you discuss very detailed in your chemistry also. So you should be clear in your mind that what is hydrogen bond, what is ionic bond, what is van der Waals forces. So that should be clear to you. Now proteins, they are also defined according to the on the basis of their functions, right? Like fibrous proteins as well as globular proteins. Fibrous, if you recall your chapter animal tissues, we said there is a substance matrix, intercellular ground substance. It is made up of a protein which is known as collagen, and it is having white fibers, yellow fibers. So this is our fibrous protein. Actin and myosin, contraction proteins which help in muscle contraction. Keratin in hairs, claws, feathers, these all are examples of fibrous proteins. Globular proteins, they are soluble in water, like you can say albumin, your egg white, then hemoglobin, insulin, then major enzyme in Calvin cycle, rubisco. These all are our globular proteins. Even immunoglobulins, our antibodies, these are globular proteins. How to remember these? There is a one word, garmi. Garmi, GHARMI. Please write down these tricks. It should be in your notes. Garmi. G for globin. G for globulin, right? G for basically globin part of, you can say globin part of hemoglobin. H for histones. Histones are the proteins which help in packaging of eukaryotic DNA. Basically, the DNA is very, very, very long. So, this is wrapped around the proteins, and these proteins are our histones proteins. Then, albumin. A for albumin, R for rubisco, M for pigment which is present in your muscles, myoglobin, I for insulin, EI for immunoglobulins, which are also known as antibodies. So in this way you can remember your globular proteins. Garmi, GHARMII, Garmi. These are our globular proteins. So most abundant protein on earth, if this question is asked to you randomly, then it is RUBP, ribulose biphosphate. Then one protein which is important as a structure wise also, and it act as an enzyme also, it is myosin. If you recall that when we draw myosin, then its head, after attaching with the actin, it start behaving as ATP ATPase, that we studied detail in the our basically muscle contraction, ATPase. So the protein which is structurally important as well as act as an enzyme, that is myosin. The last of for this protein is that if you are asked to test proteins practically, that you are given a substance and you are asked to see that if there is protein is present or not. So quantitative test for protein is xanthoprotic test. Xanthoprotic test is used to test the presence of a protein in the solution. We use concentrated nitric acid to find out the presence of protein in our sample. So this is all about protein. Go through these primary, secondary, tertiary and quaternary proteins with their examples, right? And next we will talk about lipids in detail.